This round.
Yeah, yeah.
So colleague and friend for a long time, Professor Wally Paxton from Brigham Young University. Wally and I were thrust mates, I guess, at Spin. So him from San Diego, me from the Los Alamos side there, and for about 6 or 7 years, and we were able to do a lot of my work together. And I learned a lot from working with each other there. Wally received, he actually said he had an interesting story, went from BYU. And so as an undergraduate, then to Penn State, and it's come, as you can already tell, full circle. Penn State, did the graduate work there. So BYU chemistry, right? It's chemistry.
Yeah, that's right.
Went to Penn State, PhD. Then did a couple of different postdocs. One at PNNL, which is Pacific Northwest National Laboratory. So it's his first national lab stop. Before going on then to Northwestern, where he worked with Sir Fraser Stallman, which I guess makes you a squire. Is that right?
I think I'm a squire.
Are you a squire? Yeah. Sir Fraser Stallman, he received the Nobel Prize in 2016 for his work on molecular. So he's after working with the Nobel laureate and did some amazing stuff, molecular machines, and developing a lot of technology that he then brought down to Sandia and since. Where we were for about 6-7 years, I think it's since, right? Until the alma mater called. The alma mater called and said, Wally, we want to bring you back home, and hard to turn down an alma mater, right? And so Wally packed up and moved back to BYU, and he's an associate professor there in the Department of Chemistry, and going to tell us about his work today, doing a lot of great stuff that I mean, extending beyond even the work that I knew that you did over at Cent. Still a lot of modulating surface properties and a lot of membrane and vesicle chemistry and electrochemistry. And so they were going to hear about all of that coming together in modulating surface properties and shape of artificial bilayer membrane. So thanks for coming, Wally, and welcome to NAU.
It's good to be here. I have been to Flagstaff many times before, but it was usually to get gas on our way to California. But I did explore a skate park that was down the road a little bit, which is a lot of fun. And I'm glad to spend a little bit more time here and enjoy this beautiful campus in this beautiful city. So right now, let's see if I can. There we go. So right now I'm at Brigham Young University, and this is the Benson building right here, the chemistry building, and my lab is right there, and so you can't see inside of it, but beautiful campus here. We've got 34,000 students there, and in the chemistry department we've got 40 chemistry faculty, about 30 are research active. We've got some teaching faculty there too, and about 100 grad students. And when I came to BYU to visit, that was actually something that surprised me, because as an undergraduate, I don't think I fully appreciated the depth and breadth of chemistry expertise there. And one of the nice things is that I'm rounding them out with a little bit of material science and soft matter, colloid and interface science. But it's been a great place. And we're also an NSF REU center. So we've got a lot of undergraduates that have come from different places. In fact, an undergraduate named Jasmine Genchev, who was here about 4 years ago, well, probably more recently than that, but she was in my lab about 4 years ago, and did some really nice work that was part of the research that I'm going to talk about here today. But a really great REU program that we've got there, and so I got to advertise that because we've got some really good students from that. So I'm going to be talking about synthetic models of biological membranes. So if you've taken biology, you're familiar with a cell, and you've got a cell that has this membrane, and then you've got a bunch of other things inside of there that are membrane-like and made up of lipids. And the purpose of this outer membrane is to define the cell and to control access inside the internal environment from everything else. And so a cell can control the environment in there really well by controlling things like mass transport, energy transduction, the flow of information, and can protect that cell from outside effects. And that membrane also can define the shape of that cell. And one of the things that I'm interested in is how we can incorporate and mimic the properties and functions of biological cells to create robust and responsive advanced materials, to do things like targeted drug delivery or surface modification for sensors, and creating artificial soft machines. So robotics, soft robot cells, which I think would be a really cool way to exploit some of the things that we can learn from living organisms. Now, one way to create this membrane and mimic that is by using lipids. So this right here, DOPC, it's got 18 carbon chains here, some unsaturated carbon bonds there. And these liposomes make up a wide range of membranes in biology, but they're limited by chemical and mechanical stability and limited modification chemistries, although you can get lipids with a wide range of head groups on there. But one way that we can overcome some of these issues with the limited chemical and mechanical stability is by using polymers. So one of the polymers that we're interested in using is this polyethylene oxide polybutadiene. So you've got a nice hydrophilic head group, which isn't really a head, it's sort of like a tail on that other side that likes water, and then you've got a greasy tail. And so this mimics some of the properties of lipids that allows it to still self-assemble into membrane-like structures with enhanced mechanical stability, which you can tune by controlling the molecular weight of the polymers, and you have unlimited modification chemistries for talented polymer chemists. So you can take these polyethylene oxide polybutadiene molecules, and you can disperse them in water, and depending on the fraction of the different blocks, you can create different types of structures. So you have something that's sort of a cylindrical-like structure that this can form vesicles, where the internal contents are liquid water and the external contents are liquid water. If you change the distribution of hydrophilic to hydrophobic regions, then you can create things like worm-like micelles or spherical micelles. But these things are really great. You can still form these nice vesicles. And here I've got some vesicles that were labeled with rhodamine isothiocyanate, just a fluorescent dye, so you can see it with fluorescence microscopy. Get these nice cellular-like looking things. These are the polymers. I'm not going to go through the synthesis there, but the membrane properties are substantially different than lipid-based structures. The permeability, as you could imagine, with a much longer block here, permeability goes down as the molecular weight goes up. The lateral mobility, how fast molecules can move around within that membrane, goes down as you increase the molecular weight. And the stability of these goes up pretty substantially because you have so many more non-covalent interactions that hold these molecules together because of the length of these chains. So these are really nice. And one of the other areas that we're interested in is taking some of the lipid membranes, which have some dynamic properties that are desirable, and combining them with polymer membranes, which have maybe some restricted dynamics, but some more enhanced stability. They're more robust, and creating hybrid membranes that include components both lipid and polymer. So I'm going to, yeah, go ahead.
Go back.
Yeah.
So are we to understand that M, for example, your index for the PEG or ethylene oxide, is that a specific number?
Yeah,
these
are grown by living ionization, and so these have pretty tight distributions. And so I'm going to assume when I show structures that this is going to be like 22 and this is going to be like 33. Yes, that means the number of the monomeric units that make up that block. That's right. Okay, so I'm going to tell 3 stories today. Act 1, I'm going to talk about tuning the permeability of vesicles that are composed of polymers and lipids and hybrid membranes and how we can do that. And then I'll talk about how we can control the charge of these vesicles, which can have an important effect on how they interact with surfaces that they bump into, how they interact when they bump into other vesicles, or how they might interact when they bump into cells. And then in the last part, I'm going to talk about something I'm pretty excited about, where we're using catalysis to change the morphology of self-assembled aggregates to induce micelle to vesicle transformation. We're going to use enzymatic catalysis to do that. So first we're going to talk, So first we're going to talk about permeability and transport across membranes. So if you remember from biology or biochemistry, you've got this membrane, and then you've got things on the outside, and some of those things you want to get into the inside of the cell. And there are three basic ways that you can do this passively. The first one is simple diffusion, where you have molecules on the outside, and then they can pass through here. They can diffuse into the membrane and then come out on the other side. Call that simple diffusion. Another way is you can have ionophores, or molecules that will form a complex with a charged ion. Normally ions won't get through here very easily. Got a charged, really charged structure, and that's got to interact with the nonpolar environment. The nonpolar environment can't stabilize that charge very well at all. So there's a really high barrier for getting these ions from inside or from outside to inside. But using ionophores is a way to do that. And then ion channels is another one where you create a structure that you create a pore that is specific to a certain type of ion that can cross through there. Now there's also other ways. There's activated ways of transporting things across membranes, but these are the passive ones that I'm going to be talking about here today. And so, like I mentioned before, if you've got a hydrated solute, you've got water all around this solute, and it's got to diffuse into that membrane, and then it's got to diffuse outside of that membrane onto the. And so with simple diffusion without a transporter, the barrier of that entry is pretty high. There aren't very favorable interactions between a charged species and the rest of the greasy tails of that membrane. But a diffusion with a transporter, so something like an ion channel or with an ionophore, it reduces the barrier of entry across there. So can we control transport in synthetic membranes in a similar way to how it's done in cells? Well, one way to think about this is using the solubility diffusion mechanism, which looks at the relative solute concentration. So here we're just talking about a high concentration on the outside, a low concentration on the inside. In order for these molecules to cross over, they've got to penetrate the bilayer membrane and then come out on the other side. And the concentration within that membrane is going to be really, really low. But this helps us to think about this in terms of what's called a permeability coefficient. The permeability coefficient is related to the diffusion coefficient and how thick that membrane is. Or another way of thinking about it is the flux of the molecules, how fast molecules are going across their per unit area, divided by the difference in concentration here and here. And so this permeability coefficient is a way that we can quantify and compare how permeable a certain membrane is to a particular species. So we looked at permeability and we monitored this by fluorescence. To start with, what we did is we created vesicles using, we call this a simple rehydration method. We take the amphiphiles, either the lipids or the polymers, and then we create a film that's all dried out, and then we rehydrate that by adding a solution that we want to disperse these in and what we want to be inside of those vesicles as they rehydrate. And so after that, we can do what's called extrusion, where we take all of these vesicles and we get some multilamellar structures. We push them through a membrane that has pores that are like 50 to 200 nanometers in diameter. And then we get extruded vesicles that are all roughly the same size. And then we'll take this solution and we can dialyze it to separate out all of the external components from the internal components. So if there's something that we want just on the inside, we can remove all of the extra stuff on the outside using dialysis or a couple of other methods. And then the idea here is that we would have something inside of this. In this case, we're using pyranine, this molecule right here, which is a pH-sensitive fluorophore. So at one pH, it has certain fluorescent properties. At another pH, it has different fluorescent properties. We can look quantitatively at those changes and we can determine what the pH is inside of that. That requires that we get all of the pyranine out from the outside. Okay, so these are about 200 nanometer diameter vesicles that I'm going to be talking about here. And then we take, we irradiate this sample, and then we look at this is the excitation spectrum for this molecule right here. So it fluoresces about 510 nanometers, but it's being absorbed at about 402 nanometers under low pH conditions. And then under higher pH conditions, you have an excitation peak that's around 456. And so the ratio of this peak to this peak can tell us something about the pH right there. So quantitatively, we know what the acidity is inside of those vesicles, and we know how it's changing as a function of time. We're going to do that. We're going to make some DOPC vesicles, and we're going to have the pH inside of those with the pyranine at pH 7. And then we're going to change the external concentration of hydroxide by adding something like sodium hydroxide. And we're going to look at how long it takes for the pH to change inside of there, the rate of the pH change. And so we can do that, and we follow this as over time. And you'll notice that this is the point where I've changed my base, if that's not obvious. And there's a dramatic change where the pH jumps up pretty high initially, and then it slows down. The rate of that change slows down. There's a couple of reasons for that. And I think one, and we'll talk about that in just a second. But what I can get from this right here is that from the pH is the negative log of the hydronium ion concentration, if you remember your general chemistry. But it also tells us what the hydroxide ion concentration is. And if I know how the hydroxide ion concentration is changing as a function of time, then I know the hydroxide ion flux. And that's normalized for surface area if I assume the surface area of those vesicles. From that flux, I know the concentration gradient because I know what the pH is inside, and I know what I've added on the outside. I know what that concentration difference is. I can get the permeability coefficient. And I can also get the diffusion coefficient, which is just that permeability coefficient times the distance over which that has to travel. And so that gives me an effective diffusion coefficient across the membrane. Thicker things will take things longer to diffuse across. All right, so I'm going to come back here, and you'll notice that well, you might not notice, but I'll point out that there are 3 distinct regions here. This is first, there's nothing happening here. This is where my pH has changed. There's a rapid increase, and then it kind of levels off here and becomes roughly constant there in that 3rd region. So what's going on there? Well, the first thing that happens is hydroxide ion flux is actually pretty fast. Because hydroxide ion is in water, it can travel in ways that other ions can't, right? If you have a water molecule and you've got a hydroxide ion, that hydroxide ion can deprotonate the water molecule and make another hydroxide ion. And that can go all the way down. So instead of the hydroxide ion actually moving physically, it's just transferring that charge really quickly by proton hopping. And so this means that hydroxide ion actually jumps in there really fast. That's this dramatic increase right here. But then when you get lots of hydroxide ions in there, what happens to the charge inside of those vesicles? It starts to get negative. And you have to have something else balancing out that negative charge influx inside of those. And so then that means you have to wait for some positive ion to diffuse out, or some negative ion to diffuse in, or some negative ion to move out. And so really what we're looking at right here is not the flux of hydroxide per se, but the rate of hydroxide ion flux limited by the potassium ion flux. And this ends up being about 10 to the minus 10 centimeters per second, which might not mean anything to you, but it's going to be important for some comparisons in a second. And so one of the things that we notice is that pH versus time reveals that the limiting flux of the potassium ions. But because the potassium ion flux is going to be balanced by the hydroxide ion flux, this is the same as the hydroxide ion flux. All right, so we have a way of measuring permeability of vesicles. So we did this for a range of vesicles. We looked at DOPS, which I know you can't see that right here, but this is just DOPC with a slightly different head group. And this is DOPC right here. So it's got a head group, and then it's got a couple of greasy tails right there. And then we compared this to polyethylene oxide polybutadiene membranes. And so the hydroxide ion flux, or potassium ion limited hydroxide ion flux, however you want to think about that, is about 5 times 10 to the minus 10 for both of those. And then for the polymer, as you might expect, it's a little thicker. This permeability coefficient went down a little bit. And then for a much thicker polymer membrane, so this is with 89 of those ethylene oxides, and then 120 of the butadienes, you get a even lower permeability coefficient, and then lower diffusion coefficient. All right, so anyway, the short story here is that as you increase the thickness by using polymers, you're decreasing the permeability. And we've decreased the permeability by approximately a factor of 5. We compared this to some other work that's already in the literature. This is polyethylene oxide polybutadiene, all right? This is polyethylene oxide polybutylene oxide. So instead of just a greasy tail of hydrocarbons all the way there, I've got some oxygens in there. Those oxygens can stabilize negative charges as they cross through the membrane. And so this actually is a little bit more permeable to ions than the polyethylene oxide polybutadiene by a factor of 3 different. So much more permeable. And then polystyrene polyacrylic acid. This is, instead of just the hydrocarbon here, I've got this these benzene rings that can stack on each other. And so this is going to give us a little bit less permeability for our ions. And so 10 to the minus 13. And so in addition to controlling the permeability by using polymers and increasing their length, we can also use different chemical compositions to control how fast things get across that membrane. All right, so what good is that? Well, if you can slow down how long it takes to get things inside, or how long it takes for the outside pH to equilibrate with the inside pH, then you could exploit that. And you could create an environment inside of here that is significantly different than the external environment. So pH of like greater than 10 inside of here, and then a pH less than 4 on the outside. And these gradients will persist for really long periods of time. Now, neutral species can readily penetrate the membrane. Okay, if they're slightly soluble and they bump into this, if they're not charged, then they can bump into the membrane, cross over the membrane a little bit easier. And then once they get inside of that membrane, we can do some chemistry on them and see what happens. If we make them charged species, they won't be able to get out. All right, so if I've got a neutral uncharged species, combine that with hydroxide ions, and then I make something that's negatively charged, and those won't be able to escape, might be able to visualize that. And one way that we can do that is by using a fluorogenic compound like this right here. This is called fluorescein, but it's got something different on it. So fluorescein is a really popular fluorophore. This has acetates on it. This is fluorescein diacetate. Fluorescein diacetate is not fluorescent. Okay? But if you remember your organic chemistry, we could hydrolyze those and we could create acetate ions and fluorescein. And fluorescein is going to be negatively charged, a -2 charge under basic conditions, and that would not be able to escape. So the idea is that fluorescein diacetate should penetrate through there, and then once it gets into here in the high pH environment, it should become fluorescent and light up. So we did that with giant vesicles. So after 10 minutes of this situation right here, I get a little bit of fluorescein diacetate in there. You can just barely see that vesicle, and then this is the bright field. 22 minutes, 46 minutes, and these things start lighting up. So what I've done is I have concentrated and sequestered fluorescein inside of there such that it turns on, and it turns on the fluorescence. And I can follow that over time. And here's just a couple of control experiments. This is pH inside the vesicle is 3 and outside the vesicle is 3. this does not cause hydrolysis of the fluorescein diacetate. Okay, and so over a period of time, you don't see any increase in fluorescence intensity. If I do inside the inside pH 7 and the outside pH as 7, I'm going to see what the baseline hydrolysis and fluorescence turn on is when the pH is 7. Okay, and so we do see some increases in fluorescence intensity at pH 7. But this right here is pH inside 13, pH outside is 3. The only way that this reaction can happen is if the fluorescein diacetate gets inside and turns on that fluorescence. Because this pH is too low for anything to happen on the outside. All right. You could also say, well, what if hydroxide ions get out? Could that happen? Well, if I take this solution and this solution and I equilibrate it, I just destroy all of the membranes. The pH of this solution is about 4. So it should be right here, because the internal volume of those vesicles is pretty small compared to the bulk volume. All right, and so now what we've done is we've created what I called an artificial lysozyme. It collects and digests hydrolyzable material, and then this hydrolyzable material actually turned out to be fluorescent, so we can show and demonstrate that we've actually done that. Okay, so permeability across polymer bilayers. What about hybrid bilayers? Now, you might expect we've got a semi-permeable lipid membrane with a permeability coefficients on the order of 10 to the minus 10. The polymers, maybe around 10 to the minus 11. And then the hybrid membranes, maybe they would be somewhere in between those two. So we looked at this and we created some hybrid lipid polymer vesicles. And this right here indicates the fraction of polymer. Okay, so this right here is a pure DOPC vesicle. This is with 25% polymer, 50% polymer. You'll notice what's happening to the thickness of those membranes, that you're increasing the thickness there. And so this is our 100% polymer membrane. And these are some cryo-electron microscopy images that Hera Shin produced, a postdoc in my lab. Some really nice work, and you can see the increase in size there. Okay, so we've got thicker membranes. So we might expect that the permeability might scale with the thickness there. Again, we're looking at hydroxide flux that's limited by the potassium flux, and we're just going to look at what the permeability coefficients are there. So here's the pH change as a function of time. This is when I change the pH from 7 to pH 10. And the 100% polymer, still pretty slow permeability. 100% DOPC is a little higher than that, but what was kind of surprising is that our 25% polymer hybrid bilayer was significantly more permeable than either the DOPC, the lipid vesicles, or the polymer vesicles. And so we've attributed that to a size mismatch, where because these are slightly different sizes, we've got some gaps and some defects, and they don't pack the same way that they're supposed to be able to pack in a pure lipid membrane. And so these polymers are interrupting that. So because of this size mismatch, we believe that there are more defects there that are causing this problem. Okay, so I'm going to talk about modulating permeability with ionophores. And if you remember, an ionophore is one of those molecules that will sort of cage the charge or of the ions to allow them to pass through a little bit easier. And that should increase the permeability of vesicle membranes. But we're going to use two ionophores. The first one is nigericin, and the second one is valinomycin. Both of these are antibiotics. And they do a good job of killing bacteria. And they also do a good, and the way that they do that is by allowing ions to transport that shouldn't otherwise be able to transport. So here are the results of these experiments. Right there, after a few minutes, adding a little bit of pH, changing the pH to pH 10. This is a blank. This has no vesicles in it. So as soon as I add the pH 10 solution, this should jump up. in all cases, because there's nothing to prevent the pH from changing throughout the solution. Here, this is a DOPC only vesicle. This is its permeability in the absence of any of those ionophores. But when I add either nigiricin or valinomycin, both of those completely compromise the integrity of this against the ions, and they're able to pass through practically instantly. Same thing with the 10%, polymer vesicles. So these are lipid vesicles with 10% polymer in them. And then 25%, you start to see a little change. But 50%, you'll notice that the valinomycin is not as effective. It still lets everything in after a period of time, but it's not as fast as the nigiricin. And then the 100% polymer, you can see that the nigiricin is as good as ever, but the valinomycin is not quite as effective at transporting ions. One of the things to point out is that nigiricin can transport both hydrogen ions or hydronium ions and potassium ions. The valinomycin, on the other hand, is only effective at transporting hydronium ions or H+. And so because it can only, and the concentration of hydronium ion is so much smaller now because of the change in pH, that it's much, much slower. All right, so we can control the permeability by changing the molecular weight of the polymer, by creating hybrid vesicles, or by introducing ionophores. I'm going to switch gears a little bit and talk about modulating and modeling the charge of hybrid lipid polymer vesicles. So up until now, I've been talking about DOPC and polymers that are both neutral. But what happens if I want to control the charge of this? This can make a difference in how these vesicles interact with biological systems. If I add a bunch of positively charged lipids to a vesicle, then I can change the charge of that vesicle. But would that have the same effect if I did it with polymers? And so we've got charged polymers and charged lipids and neutral polymers and lipids, and we're going to combine those to see the effect of on the zeta potential, or the potential or surface charge of these particles. So why do we care about this? Well, if you've got a couple of vesicles, there are van der Waals interactions that are going to pull these things close together. Matter likes to stick to other matter, unless you give it enough energy, and then it doesn't. In dispersed in solution, these will want to be close together, but they don't bump into each other completely because they have electrostatic repulsions. And so these repulsions kind of push them apart. These vesicles are going to have essentially the same charge, and like charges don't want to be next to like charges. And so they push each other apart a little bit. And this has a really important effect on colloidal interactions and stability. If you're going to have particles aggregate, if your cells in your body or your blood cells are going to coagulate or something, and that's supposed to happen outside the body, but not inside the body. And one of the reasons for that is because of these different ways that charge is mediated and other intermolecular forces are mediated. This can make a difference also for how things adsorb onto surfaces. So vesicles can adsorb to surfaces if you have the right balance of attractive interactions between the vesicle and the surface. So you could change the properties of an interface by adsorbing things to it. And the same thing with cells. If you have vesicles that are a particular charge, that electrostatic repulsion or attraction can mediate how these are going to interact with each other. Now, this is really important. This is important because in 2020, we had this global pandemic and everybody was trying to figure out, how are we going to make sure that this doesn't happen again? Or how can we protect people from this? And one of the ways that they did it is that they used lipid nanoparticles that are cationic. They're positively charged. And inside of those lipid nanoparticles, they have genetic information. And that genetic information, that RNA, codes for some target or the spike protein that will teach your immune system how to resist COVID-19. And so one of the strategies was using these lipid nanoparticles to do that. And so this is incredibly important for how you can get payloads into cells. So we care about vesicle charge, and other people do too, it turns out. So one thing that is another important aspect of vesicles in the body is that you don't want these things to be cleared out from the body too quickly. Okay, you don't want to get a COVID vaccine that your body immediately clears through the system. And so one way to prevent that from happening is using polymers like polyethylene oxide as a steric stabilization. Basically, these polymers will prevent opsonins from interacting with this and marking it for being cleared from the body. Okay, so if the immune system can't mark this, then the immune system won't know to clear it out. And so these have longer lifetimes so that they can deliver the payload that they need to deliver. And so with even 2 to 10% PEG, you can get reduced biofluid interactions, that they don't interact as well. You don't get the marking by the immune system for clearance from the body. So how does PEG affect the charge on lipid vesicles? Well, if you have a lipid vesicle like this is a negative 10 millivolt. So this symbol right here means zeta potential. This is just a measure of the potential of that surface. And so as you might expect, negative is a negative potential. And this is just the magnitude of that. But we call that zeta potential. And so you'll see that throughout on some of these slides. So this zeta potential of this particle is negative 10, which means it can bump into a positive surface and it would be attracted. But what if we took this negative 10 millivolt zeta potential particle and we added a little bit of polymer onto the outside of that? If you add even 10% of PEG lipid, you'll completely screen the charge of that. This is going to make it really hard to change the charge, the surface charge or zeta potential of polymer vesicles, because you can't just add negatively charged lipid to a polymer vesicle and change its charge. Yeah,
Okay.
yeah. So these are just, yeah, exactly right. This is, I'll show on the next slide, but it's DOPS and DOTAP is the other one that we use. So we're going to modulate that polymerisome charge by adding charged lipids. So if I add cationic lipids, what happens to the zeta potential? Anionic lipids, what happens to the zeta potential? So we have a range of lipids that we can use. So DOTAP, yeah, this is the positively charged one. We've got so this shows your polymer and your lipids, and that head group of those lipids can be either positively charged, neutral, like with DOPC, or negatively charged with DOPS. Obviously, they have much longer names, but I'm just going to call them that because it's for simplicity. And then on the other side, we've got polymers, and the end group of these polymers is just a hydroxyl group, which is neutral. Okay. So how much of these would I need to add in order to change the zeta potential of a neutral polymer vesicle? So this was some work done by some undergraduates in my lab, Trent Johnson and Sydney Johnson. How do we change the charge of this neutral polymer zone? Okay, so we got DOTAP, DOPC, and DOPS. So these are the zeta potentials of the 100% DOTAP vesicles, 100% DOPC vesicles. And this is what we observe. This is the fraction, the mole fraction of polymer that we've added to the composition of these vesicles. And you'll see that the zeta potential drops off pretty fast. Okay. If I added 10% of charged DOPS to these vesicles, I'd be right here. And that's exactly the same as the zeta potential of the polymer vesicle all by itself. Okay. So I've got to add a lot more. Even at 50%, it's not until we get to like 60%. So, well, let's see, 70% of the composition has to be the lipid in order to start seeing that charge. So you have to add a significant amount, way more than you ought to, in order to change the zeta potential of these particles, which will change how they affect. And so at that point, you're really just working with a lipid vesicle here. And you don't have really any of the properties of the polymer vesicle. Now, if we want to keep the polymer vesicle and change the properties, change the surface and retain the properties of and robustness of the polymer vesicles, we've got to find another way to change the zeta potential. So Keith Willis, a graduate student in my lab, looked at this problem and he said, well, maybe we can model this. So we looked at the zeta potential of a hybrid vesicle. We can predict the zeta potential of a hybrid vesicle that's composed of polymer and lipid. And so we look at the fractional area of the lipid and the fractional area of the polymer, and that this zeta potential is going to be some linear combination. But because the polymer is a little bit bigger, it can screen those. And so, so we add this extra exponential term to account for the fact that a polymer, let's see, that a polymer that is much larger than everything around it can still screen a pretty large area. And to demonstrate kind of what's going on here, this nice little demonstration. So this is all lipids, and then each one of these dots, yellow dots, this is replacing a lipid with a polymer that can screen more around it. And so you can see that these will cover up a pretty large area. And then what happens when you get one right next to each other is that it covers the same area. And so that's one of the reasons for this exponential right here. And so if we follow this animation, start replacing, I just want to point out that at about this point right here, half of the surface is replaced. But you don't see any of the lipid, you only see the polymer. And so we modeled that, and so this is our fit to the data with this model right here, and it fits pretty well. The difference is that S parameter right there, that's the degree of screening, how easily it is for something to screen. Yeah, Gabe.
Like in the model?
We did not look at it in this model. We assumed everything was pH 7.
Okay.
But we do look at that a little bit later with some of the polymers. Yeah. And so this S, we called that a screening parameter, and the screening parameter for this for the DOTAP is actually a little bit higher than the screening parameter for the anionic, and part of that might be due to the size of the head groups of the DOTAP and the DOPS. But we can predict what the zeta potential will be now if we know something about the properties of the lipid and the polymer and the composition of those two. Okay, so I just showed you how we could change the zeta, we could change the lipid for either cationic or anionic, and that can change the zeta potential of these vesicles, but it has to be a lot of that done. You have to add a lot of that. And so another strategy is by changing these end groups here, we can also change that zeta potential. And so this is the zeta potential of our, over here, this is 100% of the hydroxylated vesicles polymer, and then up here this is amine terminated, and this is carboxylate terminated. And you can see as I increase the fraction of the modified polymers, I can change the zeta. Not a whole lot, but I can change it. And so I'm gonna skip over this well, this is important to explain. So we did this experiment where the total polymer composition of each of these vesicles is 50%. The 50% was lipid. The total charged amphiphile was 40%, which means that I either had 40% of the lipids were charged, or 40% of the polymers were charged, or any combination in between. So 20% lipid, 20% polymer. And that's a function of the location of the charge group. So over here, all of the charge groups are on the lipid. And over here, all of the charge groups are on the polymer. And so as we increase the fraction of charged groups on the polymer, we increase the zeta potential of those particles. And then this actually ended up being pH dependent. Okay, so I've got this polymer, and under really acidic conditions, it's protonated, but the acid is protonated too. And so it doesn't matter if I increase the amount of acid, it won't change the zeta. And then at pH 12, all of this is deprotonated. And so I don't increase the zeta potential. We actually see a little bit of a decrease there. And we're not sure why that is, but in any event, at high pHs, we change that zeta potential. Let me just get a quick check of time. Okay, all right. So real quickly, polyethylene oxide polymers can screen the charge of lipid vesicles. Those are the stealth hybrid vesicles. And we can also, the short story is that we can modulate the charge, and it allows control over how these structures are going to interact with other surfaces and how they're going to interact in biology. Okay, so I wanted to talk this third one. This is artificial morphogenesis of stimuli responsive materials. We're going to change the shape of membranes now, and there's some reasons why we want to do this. If we could control membrane structures, we could control how they interface with biological organisms. If we could create membranes that could communicate either by taking in signal from outside, or by releasing something from inside of the vesicle to outside, or artificial actuators. If we have membranes with sufficient mechanical robustness, then we could have phantom hands that come out and grab things. And we thought that that controlling those membranes might be an important step to making artificial soft robotics. And so there's a couple of ways that we can change the curvature of different structures. One of those is by taking a membrane and adsorbing something onto the outside of it, something that sort of crowds out, and you attach enough things around it and they start to want to be further apart from each other. And one way that they can do that is by making these giant protrusions that will project outside of these vesicles. And so here's a really nice image of that happening from Jeannie Stakowiak's paper. This is membrane protrusion driven by a pH gradient. So here is a vesicle, and then this right here is a pipette, and you can infuse a little bit of hydroxide ions from there, sodium hydroxide, and this membrane will actually change in response to that chemical signal. So changing its shape in response to a pH gradient. And then even something as simple as a micelle to vesicle transition. If you have certain types of surfactants, they can either favor the formation of micelles or vesicles. And these have been characterized previously in a nice paper by Ederth and Harik. But in all of these cases, that the stimuli is externally imposed. This is externally imposed chemical stimuli that's causing those changes. And we'd like to have something that's a little bit more internal. And so we have some experience with changing the shape or changing the structure of molecules that self-assemble. And this is chemical communications that we published in 2018, where we use polyethylene oxide polyacrylic acid. At low pHs, these will form vesicles. The negative charge here is shielded by the proton, and so these will self-assemble into vesicle structures, which can be destroyed at higher pH as we deprotonate all of those carboxylate groups, negatively charged structure, and then that completely falls apart. And so we demonstrated this. This is a nice movie that a summer student, Paul Baker did. And so this is a pH 2 solution, and he added base to it. And these are the polyethylene oxide polyacrylic acid vesicles. And this is a real-time video, so it's going to take a little while. So, but keep your eye on that and also pay attention to this. So this is Alexa Floor 488, which we used as a cargo. And we put that inside of these vesicles. And so this is just a surrogate. We can see this really easily, but you can think of this as maybe being a drug molecule or something that we put inside of here. This is our polymer. Oh, you can start to see these starting to fall apart now.
It's real time.
So this is real time. Yeah.
Honest. So
I would scroll it faster, but my computer crashes when I do that in PowerPoint. So you'll get to see these things completely fall apart now. So those vesicle membranes, catastrophic failure. And so if we take these vesicles and we put Alexa Floor 488 inside of them, we can look at them. We can look at both channels, the green and the red, and at very low pHs that are raised to up to pH 6, you start to see leakage of the cargo and then catastrophic failure. We increase the pH a little bit more, leakage, catastrophic failure, and we can follow this by time, and we can look at rolling the rate at which these leave by looking at the fluorescence intensity. Now, one thing that's great about this is an enteric coating or an enteric material, where at low pHs, like in your stomach, these vesicles would be completely intact. And then at high pHs, like in the duodenum or down in the small intestines, the pH is 7, these things would fall apart. Could allow things to survive the harsh digestive environment of the stomach. Yeah. Alexa Floor 488, this is just a fluorescent dye that we're just using. So that's the green that you're seeing there. Yeah, but you could imagine putting anything in there.
Well, when you change that pH, you immediately change it to like whatever, you go to 8, and it still takes 10 minutes for that thing to fall apart.
Yeah. So one thing that you have to think about is that this is not just one carboxylate.
Yeah.
It's like 50 or 100.
Yeah.
And so you actually have a little bit of buffering capacity of this.
Have you looked at changing that PAA length and the impact?
Yeah, we've changed the PAA length. I'll have to double check and see in the paper if we quantified how long it would take for this to happen. But yeah, you'd expect that a longer chain would take longer to fall apart.
Yeah, and that's a cool thing, right? If you can get the time release.
Yeah.
Dependence, right?
Yeah, where you could control that. Yeah. All right. So moving in a slightly different direction, we wanted to look at pH switchability of these of some different supramolecular aggregate structures. And so we use this molecule MSDH right here. It has a longer name, but I'm just going to call it MSDH. And it can be either protonated or deprotonated. And when it's protonated, it has a slightly larger head group. That positive charge is going to interact with other positive charges around it. And so it gives us a little bit more of a wedge shape. And at really low pHs, where this is protonated, you get micelles, really small structures. And when it's protonated, or when it's deprotonated, then it adopts this cylindrical structure and much larger structures there. And these have been looked at by cryo-electron microscopy. But what I wanted to talk about is how you can switch these with pH. And so, but the way that you supply that pH change can make a big difference in how it, in the kinds of structures that it forms. And so if I take MSDH micelles at a really low pH, so pH 5 or so, and then I externally impose, I manually titrate in a little hydroxide, then I can change the molecules from the protonated to the deprotonated state where they favor the formation of vesicles. But what if I supply that in a different way? Instead of titrating in some molecules, some hydroxide, and mixing it up real good, what if I did something internally generated, using something like a catalytic reaction? A catalyst is dispersed throughout the solution, and it can create a change in pH catalytically that happens uniformly throughout the whole solution without me having to mix anything. And so in this case, this is substrate activated. So whatever substrate I add translated into a change in pH, would we get the same thing or would we get something different? All right, so I'm going to go a little bit quickly through this, but basically we had MSDH and we had a little salt solution and we had a little Texas Red DHPE so that you could image what was happening. And then we added sodium hydroxide to this and mixed it up real well. And so here are the results. This is the apparent average, the size of those aggregates as a function of time and as a function of pH. So here, this is like pH 5.2. We get here, this is about pH of 5.8, where we start seeing significant aggregation. And then when we get beyond, when we get to like pH 6.3, then these all collapse back down to some smaller size structures. And we looked at these by fluorescence microscopy. And so you don't see any micelles because they're too small to be resolved with the fluorescence microscope that we had. But you see bigger aggregates right here, which we're attributing this to the pKa of that acid. And then they collapse down to some smaller structures, but we're still seeing things in the microscope there. So we did the same thing, but in this case, what we wanted to look at was using urease and urea to generate CO2 and ammonia. The net change is an increase in pH that can be really predicted, or just predicted, I guess. So MSDH at pH 5 and MSDH at pH 7, we might expect to see vesicles there. We did the same thing. I love this plot here because it shows the real gentle increase in pH, and then the size of the aggregates in a completely undisturbed solution, really large aggregates, and then coming back down. So qualitatively, it looks very similar to what we saw before. And then we looked at these three different regions before. We don't see anything. On the peak, we see some large aggregates. And this is one of my favorite pictures here, is that you can see that even though it's saying that we've got 200 nanometer diameter structures, we've actually got these giant unilamellar vesicles. And to really drive home the point, my graduate student, Nick Baer, did some cryo-electron microscopy. These are externally imposed MSDH pH changes where we get stuff. And then here are the giant vesicles that are formed by catalytic activation, which was a really nice way of making giant vesicles from something that previously the largest structure you could get was about in diameter. So that was a really nice advance. So we've got a pH responsive surfactant. One of the things that I like about catalytic pH changes is what it did is it translated a chemical signal that the aggregates didn't care about, urea. They don't care about urea. But what they did care about is the pH change that occurred when you used urease to hydrolyze it and generate ammonia and change the pH. So we can translate a chemical signal that the aggregates don't care about into one that they do care about to make larger structures. So this is catalytic activation in aqueous phase systems to activate responsive membranes to different chemical signals. Okay, so I've talked about these three different areas, and I'd be happy to answer some questions. I just need to show quick acknowledgements. A lot of people in the lab from Sandia and from BYU who have made this work possible. And then I just want to do a quick commercial for this podcast that I do that I actually featured Gabe on over the past year. And we just talk about what soft matter scientists do and how they got started. And one thing that we have learned from that is that there are no direct career paths anymore. Everybody seems to take a different direction, and it's been really fun to hear everybody's different stories. Thank you guys for your attention. No, we have not. What we've done is we've said hybrid vesicles are a lot more leaky than we thought. Why? And so we dug into the literature, and there's a paper by Paul Beals who I can get. Let's see, it's kind of a ways back there. I don't know where it is. But there was a paper where they attributed that they pointed out that there are significant defects when you have mismatched molecules that are trying to be right next to each other. And in some systems, what that results in is complete phase segregation. You'll get lipid and polymer. In this case, they're similar enough that they want to be next to each other, but not so similar that it doesn't cause problems for permeability.
Let's jump in there real quick, Nico, for your next one, because I was wondering, how have they looked at like localized 2D diffusion of those, even within those types of phase segregated areas? That could be leading to that as well.
No, like a raft or something.
Yeah, exactly.
Okay.
No,
but we did some fret studies. I didn't talk about them here, but when we made hybrid vesicles, it looked like these were homogeneously distributed throughout with our polymers and with the lipids. So we didn't see any phase segregation at all.
thanks. Go ahead, Nico.
So, also I forgot to say, thank you for your talk, it was awesome.
Thank you.
And so the second question is a big question about how you approach it with your students, because it seems like, and this is probably in general in science, that you have a lot of different components of your research that build up to other ideas. So my question was, how do you approach those areas where nothing goes right? Like when you're troubleshooting, how do you approach that with yourself or with yourself and your students?
So this is a great question, because what you end up seeing in a talk is sort of the highlight reel. And what you don't see is all of the things that didn't work. And the way that we approach is we just try something different and we just keep going. I remember a quote from Chemical and Engineering News. It said, being a PhD means that you can get up every day and be super excited about things that you know almost certainly won't work. But the things that do is why we keep doing it, because they do. And if you keep pushing, you start answering questions that nobody else has been able to answer. We talked about that a little bit before.
Being a baseball player, hit 300, kicking butt.
That's right.
Other questions? Go ahead.
You talked about how there were some of these the spheres that could grow like a spike. And I was wondering what would be the application behind that design. Like I saw that there was like a red sphere and a green arm that kind of poked out. I'm curious to see what.
So they were looking at changing morphology and looking at ways to interface other cells. And so if you could imagine some cell or some vesicle, and then you create a protrusion, and then that protrusion attaches to another cell, you could interrogate what's happening or communicate in some way. And so one of the things that they were doing was trying to control this so that they could do that.
Really interesting point. So I'm curious, so oftentimes the lipid characterize lipid flipping probabilities or and it ends up being an important metric for understanding protein stabilization. You looked at your artificial lipid flipping between the leaflets? Because I assume it would be way slower, right?
No. I think that someone has looked at, we don't have the tools to do that, but there's people like John Conboy at University of Utah who looked at lipid flipping, and they've looked at fatty acids are way faster than lipids. And these polymers here, because there's no charge on them, they actually might be faster than you might think. But there's a serious, the probability of getting that whole chain to go inside of there is maybe a little bit lower than a lipid head. I'm just kind of thinking this in real time. I would say that yes, it's possible. It might have been measured, but you're right, it's probably slower.
From the cartoon you showed where they cover this huge area of the surface,
Uh-huh.
that makes it seem like they probably are more laying flat across it.
Yeah,
So then you have this kind of steric process where it has to worm its way down.
yeah.
to get in.
But then when you make 100% polymer bilayer instead of laying, they're all right next to each other. And now this thing has to move down
Yeah.
against some pretty significant steric barriers in order to get to the other side. But I guess the way to do that would be like probably get some deuterated forms of these polymers at a significant price.
Yeah.
and then measure that, because there's some ways to do it with SFG to look at how that might equilibrate.
Yeah, I think people have done it with tripod copolymers.
Yeah.
Uh-huh.
They functionalize both sides differently.
Uh-huh.
And then there they have also the advantage that they don't necessarily have to just be linearly, right? So they can form like a U shape,
Uh-huh.
and that U shape can kind of be like the intermediate between that flipping process. So
Yeah.
it's not just a complete flipping.
Yeah, that's right. That'd be a good candidate for measuring that. Yeah.
Just a quick question, have you tried to measure the theta potential after a certain time, after a long period of time, maybe at the end, you have a specific theta potential that you show, maybe after 2 months or 6 months?
This is a great question. And I think that, and this might not be a satisfying answer, but the lipids themselves are not very stable. And so over the course, we won't use lipids after 1 week. If we use them within 1 week, then we can do this. And so I think most of these were done within a week. We didn't notice any changes over that time scale. But after that, once you start like pulling off some, you get hydrolyzing some of these, or you start oxidizing those double bonds in the oleal tails. That's hard to say. Then it starts to complicate things a lot, and you get things crashing out of solution. And so then it's all bets are off. Yeah. Good question though. We would love to have a more stable system, because material science, I mean, in the body, this is no problem, because it's like, no, like a lipid died, no big deal. We'll just make another one. But in a synthetic system, when you're trying to do material science with this stuff, the stability of these things really is an issue. Yeah.
Great. Did you? Okay, last question.
This one. So we were talking about the pH induced disassembly. Is there a certain rate you're looking for? Do you want it to be faster or slower, or are you just kind of looking at the process?
We just wanted to see if we could control it at all. And so, as I had a professor once say, the wonder of the singing dog is not how well it sings, but that it sings at all. And I think that this is because we could tune that, and we could. But one of the interesting applications we were thinking of is if you have an enteric coating, then you could do oral protein delivery, which you can't do right now. Diabetics have to inject themselves. The reason they can't eat insulin is because their body says, oh, it's protein. So it just digests it. And before it can actually do anything useful.
Yeah.
Hey, just a real quick comment. I mean, great talk, and I have 100 questions, but I just want to point out, Jasmine Genchev, who you mentioned, right? And credited as your summer REU student from here and AU. She graduated last summer. There's a photo of her up on the 4th floor. I think Eric could correct me, but I think that was the top prize for a chemistry undergraduate research. She worked with Dr. Naomi Lee here in the virus-like particles. And you pointed her out at the beginning of the talk, and then you had credit at the end. So I just want the people here to hear that their teaching of the undergraduate students makes a big difference.
That is a really important connection to make, is that your work with those students makes a difference for them. Yeah, absolutely. She's fantastic. She's at Northeastern University in Austin now.
Continuing in
Yeah.
the virus.
Yeah, I try to get her to come back to BYU. You know,
She
Northeastern is a
it's hard to turn
fantastic
down, yeah.
place. Yeah.
All right, let's say this takes a long